Everyone. So welcome to today's BRAIN key lecture. Our today's speaker is Michael Belloy from Stanford University. And as usual, we will have a talk about for 30 to 40 minutes, followed by a question and answer session. So please come forward and just unmute your microphone and activate your video in order to ask the question. If you do not want to do this in person, just write me a text message via chat and I can ask the question for you. So now I'm very happy to introduce Michael Belloy. He is a postdoc at Stanford University in the lab of Michael Gracious. And he has been working there for 2 years now. Before that, he got a PhD in biomedical science from the University of Antwerp. So today he will talk about Klotho. We have learned recently that rare genetic mutations like the Christchurch mutation can significantly modify the risk of the APOE4 genotype. Klotho is a more common variant, which has also a significant effect on the risk of Alzheimer's disease. And Michael's study is probably the most convincing study. He really collected a large amount of data that are supportive of the protective role of Klotho in Alzheimer's disease. And I'm very much looking forward to your talk, Michael.
Thank you for the introduction. Just bring up my slides here. Just to confirm, you can see the slides. It's good?
It's fine.
Fine. Oh, cool. All right, thank you for the introduction, and also thank you for inviting me. It's actually the first time I'm being invited to present, so that's very nice, and I'm also very happy to contribute to this keynote series. So indeed, today I'll be talking about Klotho VS heterozygosity and how it reduces risk for Alzheimer's disease in APOE4 carriers. And so before I start with that, I want to give like a brief background on the genetics of Alzheimer's disease, which is the main focus of our lab. It's the main question we're trying to work on, and we're trying to disentangle essentially the genetic architecture of Alzheimer's disease. And so what I presume that most of you are probably aware of is that the most prominent or the most strong genetic risk factor for Alzheimer's disease is APOE. And so if you look here on the left, we know that if you have 2 copies or 1 copy of APOE2, of the APOE2 allele, you have lower risk for Alzheimer's disease. And if you have 1 copy, and particularly if you have 2 copies of APOE4, you have substantially increased risk of Alzheimer's disease. And so this is something we already know for a long time. We know this for more than 2 decades. And so over the last 2 decades, there has been large and very fruitful, large-scale studies that have been focusing on genotyping common variants across AD cohorts. And what this allows researchers to do is to run genome-wide association studies of Alzheimer's disease. So for instance, here you see the most recent, largest genome-wide association study of AD, where we see here a Manhattan plot, and on the x-axis we have all the chromosomes and increasing base pairs covering different variants, and on the y-axis we see the minus log 10 of the p-value for a particular association of a given variant. So for instance, a value of 50 here corresponds to 10 to the minus 50. And so what you can see on this plot is that there are like a bunch of little towers sticking up above this red line, which we call the genome-wide significance threshold line. So essentially all these little towers, they show you regions or variants that appear to be significantly associated with Alzheimer's disease. And so this is very important because they teach us about the molecular etiology of Alzheimer's disease, and they help us target particular pathways that could be relevant to new drug design. And so what stands out here on chromosome 19, which is APOE, and we know that already. And actually this plot is kind of also skewed. It's not really representative because it's being capped off at 50. If we actually take a look at how the plot really looks like, not capping it off at 50, we really see that APOE towers massively above all these other different variants. And so now that I've shown you this, I have a poll question here for you. So if I would tell you that 53% of Alzheimer's disease is genetically inheritable, how much do you think APOE is explaining, And how much do you think these other genetic risk factors that have been discovered over the last 20 years are explaining? So there's 4 options that I put here.
So if anybody would like to respond, please do it now. Otherwise, we will close the poll. Okay, thank you very much. And here are the results.
All right, so most people are saying APOE 20%, other variants 10%. So at least the pattern is clear in that it's true that it's APOE is explaining more than the other common variants, but actually the difference is much more pronounced even still. It's actually so that APOE is explaining a very significant fraction, and these additional variants are only explaining a small additional fraction on top of that. So close that. All right, and so this is actually a visual overview of that if we look at what we call the missing heritability of AD. So this is based on a study from 2016 that estimated that based on genome-wide association studies, 53% of AD phenotype can be explained through genetics, but only in total 16% is currently attributed to known variants, but more than 30% is still, we don't know where it's coming from. We haven't tagged particularly any specific variants that are making up this missing heritability. So the particular aim of our lab is to disentangle what's this missing heritability. And so the way we approach that is mostly by statistical interactions. So most prominently, we're looking at genetic variants that interact with APOE4 to change risk for Alzheimer's disease, depending on APOE4 status. We can look at interactions with age. There is more and more literature supporting that the genetic associations with Alzheimer's disease will differ across age. We also look at non-additive models. So most of these genome-wide association studies, they look at effects that are increasing with the amount of alleles a variant has. So 0, 1, 2 would be a stepwise increase or a stepwise decrease, but there are other type of genetic models like the one we will cover later. And then we can also look at interactions with ancestry or interactions with sex. But to keep it a bit more simple for the focus of this talk, we're going to look at these 3 interactions between APOE4, age, and non-additive models. And particularly, like I mentioned, really our main question in the lab is, why do some APOE4 carriers remain asymptomatic? Are there particular genetic variants that they have that make them be protected? And so our framework for answering this question is we try to acquire and curate as much large-scale genetic data as we possibly can. The same for functional validation data. So anything going from A beta PET, tau PET, to structural imaging, to cognitive scoring, to protein expression levels, and so forth. We try to gather as much data as possible that can help us validate any variants that we find from large-scale gene association studies. And then we try to develop hypothesis-driven questions and collaborate with other people within and outside of Stanford to find new gene variants. And so, although I'm for sure not an expert on cognitive resilience and resistance, I try to put together kind of my view on how this is part of our work. And so when we try to identify these variants, most prominently, we're essentially interested in those that give resistance to amyloid and tau pathology. But we're also interested in variants that provide resilience, so they allow you to cope with pathology. One way we can look at that, for instance, is if we have subjects that we have amyloid status and we know subjects are amyloid positive, how does the genetic variant that we're investigating affect cognition or brain structure or function in those subjects? And then reserve, to some extent, is also part of our work in the sense that where we have it available, we'll adjust for the amount of years of education a person has. And there may also be genetic contributors that are increasing reserve and by such are reducing risk for Alzheimer's disease. And so I'll focus on one particular interaction right now as a last part of this general background. So this is how APOE4 and AD interact across age. And so here again, I have a question for you. What do you think is most appropriate here in terms of when APOE4 is conferring risk for AD, do you think this is increasing with increasing age? If it's higher at 60 to 80, 70 to 90, or 80 to 100? So maybe we can pull up the poll. Yeah, great.
Okay, a few more seconds. Please make your choice. Thank you.
Yeah, exactly. I think most people are indeed knowing quite well the literature already indeed. So it's true, APOE4 has the highest risk conferring effect on Alzheimer's disease at the ages of 60 to 80. And so just to give you some visual illustration of that, for the data that we have actually access to from different public resources, so over 20,000 subjects, we see here a sliding window analysis of risk for AD due to APOE3/4 between the indicated age ranges, and you really can nicely see that around 60 to 80 we have like a strong increase relatively compared to older ages. And for APOE4 for subjects, it's even earlier skewed, and of course much more strongly affecting the odds ratios. And so if we look at it in a more dichotomized way, which is also more established in the field, we have this difference between the effect of APOE4 and AD between 60 to 80 and 80 plus, and essentially by 80 plus, the effect is halved compared to 60 to 80, being an odds ratio of about 6 here. And so this effect on AD is actually something that also relates to APOE4's effect on amyloid pathology, right? This is the most prominently known feature of APOE4, that is increasing amyloid pathology. And so we similarly see that in pre-dementia phases, so people who have normal cognition, subjective cognitive impairment or mild cognitive impairment, we see that during this age range of 60 to 80, subjects with APOE4 simply have generally higher levels of amyloid pathology, and they also have steeper increases in the amyloid accumulation. And once you reach the age of 80 plus, you start to have more plateau effects, essentially. So overall, genetic associations with AD seem to differ across age. There is some literature recently supporting that, and particularly associations of APOE4 with AD and amyloid pathology also differ across age. So because of that, we essentially hypothesize that genetic interactions with APOE4 that mitigate risk for AD will also differ across age and across these different age ranges of 60 to 80 and 80 plus. So now moving forward to Klotho. So Klotho protein, you can see it here on the left, that gets actually cleaved at the membrane and gets shedded as a hormone into the surrounding tissue, in the surrounding space. And so Klotho is expressed in multiple sites in the body, so it's present in the serum, with the main source being the kidney. It's also present in the CSF, with the main source being the cortical plexus, which actually creates relatively high levels of Klotho. And also in the central nervous system generally, neurons appear to be creating and secreting Klotho. And so Klotho in of itself also has like a very extensive literature showing that it affects many different pathways, including insulin signaling, fibroblast growth factor, and so forth. And where it's really the most known for is that it's supposed to be a longevity gene. So some studies have, the initial studies actually showed that increased Klotho levels promote longevity and decreased Klotho levels decrease longevity. And so with regard to healthy cognitive aging and resilience, it seems that there's actually a large variety of studies suggesting that Klotho levels are improving synaptic function, enhancing cognition, and also enhancing cognitive resilience and brain aging resilience. And beyond that, some other smaller studies that are also very interesting suggest that Klotho levels are also decreased in subjects with Alzheimer's disease compared to control subjects. So here on the right, you see this small plot showing you Klotho levels in the CSF for 20 control patients and 20 AD patients, and showing you how this actually differs significantly between them. And also we know that with increasing age, Klotho levels go down in CSF and plasma, suggesting that this might be a link towards seeding, with increasing age, increased risk for Alzheimer's disease. And what's very nice across all these studies is that they're supported by a variety of both rodent and human studies, and both studies looking at samples in serum and CSF. And so now about Klotho VS, which is really the main point of this talk, or the centerpiece of this talk. So Klotho VS is a functional haplotype. And so a haplotype essentially means a combination of variants that are in linkage to disequilibrium with one another. So what that essentially means is that if I have variant A on the left, if I know what allele this one has, I always know what variant allele, which allele variant B has. And so these two variants together both are affecting amino acid substitutions. And so the substitutions that they affect are called V and S. So that's why we actually talk about Klotho VS. And what's particularly interesting here is that it appears that one copy of this haplotype, not two, increases Klotho secretion. And with this effect we call heterozygosity. And so to give you here a visualization of that, this is from Dubal et al. From 2014, and later also Yokoyama, showing you that Klotho serum levels appear to be higher in Klotho heterozygotes compared to non-Klotho heterozygotes. And so other interesting things that have been shown for Klotho VS heterozygosity is that it's associated with increased longevity, better cardiovascular health, and increased cognitive ability and reserve. And most particularly, what really got us involved in this is the 2019 study by Erickson showing that Klotho heterozygosity was attenuating amyloid burden in APOE4, in preclinical APOE4 subjects. And so here on the right, I show you essentially the main findings from their study, looking at amyloid burden and showing you the t-value for the APOE4 related effects on A beta positivity in APOE4 negative subjects, where you see a strong significant effect, and in APOE4 positive subjects, there is very strongly decreased effect, and there's no longer a significant effect, suggesting that APOE4 is no longer conferring its deleterious effect on amyloid pathology. And so because of this study, we essentially wanted to ask the question, what's the role of Klotho in Alzheimer's disease? So yeah, like I mentioned, we want to know, is it protecting against Alzheimer's disease? And specifically based on this prior study, we hypothesized that the effect of Klotho heterozygosity would be specific to APOE4 carriers. And so to tackle this question, this hypothesis, we built three aims. The first one was to evaluate this question in APOE4 stratified analysis in case control data, and then to have essentially asked the same question in conversion analysis, so evaluating if subjects are more or less likely to convert to MCI or AD. And finally, also to try to replicate this effect on A beta burden that was observed in the prior study. And so what we did is we put together data from 25 publicly available AD-related cohorts. And I'm not going to go into detail on that, but what we did is like do all the standard genetic data, quality control, and harmonization. And then we restricted our analysis to subject of the largest and most homogeneous ancestry population, which is Northwestern Europeans. This is essentially making up, I would say, about 80% of the data that we have. And here I give you like a small demographics overview of for which analysis, how many cohorts were included in there, and how many subjects were included in there, what kind of age information we're using. And so what I'll point out specifically here for the case control data, the age values that we have for AD subjects are age at onset. So this is when the clinician asks the patient, at what time did you subjectively start to experience cognitive decline? This is not the same as the first clinical diagnosis of dementia, for instance. So between these two, there might be about a 10-year difference, for instance, on average. And so in terms of analysis for these different types of questions, overall, the study was always, for any analysis, had a meta-analysis design. We also did mega-analysis as a sensitivity analysis, and the effects are always concordant. And then for the different type of models, so if we look at case control data, we use GLM-based approaches. If we look at conversion risk, we look at competing risk regression, where we use that as a competing risk factor. And for the A-beta work, we look at just basic linear modeling or linear mixed modeling when we have access to longitudinal data. And so this is the main first finding, suggesting that Klotho VS heterozygosity is reducing AD risk in APOE4 carriers specifically. So here we look at the full sample, being all subjects above the age of 60. Here you get to see broken out per APOE stratum. You see the amount of cases and controls in the analysis, the amount of heterozygote carriers across them. And then you see the odds ratio and p-value suggesting about a 25% reduction effect. And so at this point, I'll put my last question, my last poll question is, what do you think is going on with Klotho VS heterozygosity, how it is reducing AD risk in APOE4 carriers across age? So do you think it doesn't matter what age subjects are? Do you think it's more strongly at 60 to 80, at 80 to 100, or if it's actually increasing risk between 60 to 80?
Okay, please make your choice. Thank you.
All right, so I guess I gave hopefully a relatively decent introduction. Indeed, the effect is more stronger at ages 60 to 80, and that's what we would expect if there is an effect on amyloid pathology. So if we take a closer look at the actual results, if we look here, we have the age stratum 60 to 80 and 80 plus, again, broken down by APOE4 status and showing you here the odds ratios. So if we look at 60 to 80, we see now actually almost a third decreased risk for Alzheimer's disease. The significance has gone up further. And this actually, in terms of genome-wide association studies, would also qualify as a genome-wide hit because it passes the 5 × 10⁻⁸ threshold. And what's also quite interesting here is if we look at the 80 plus sample, despite the fact that we have lower power, obviously, because the sample size is smaller, we see no effect at all of Klotho VS heterozygosity, really suggesting that the main effect is strongly restricted to subjects that are 60 to 80. And so just to give you a sense of the heterogeneity or lack of heterogeneity across cohorts, we see here that the effect in APOE4 carriers was really very concordant across the different cohorts, with only one cohort showing a different effect. And if we look at how the APOE4 interactions look like, so essentially asking the question, is it really specific to APOE4 carriers or not? We also see that this actually fairly well holds up across the different cohorts included in this analysis. And so now that we looked at case control status, now we're looking at, then we looked at conversion risk, so conversion risk to MCI or AD. And on this plot, you see here in blue the subjects that don't have Klotho VS heterozygosity, and in red the subjects that have Klotho VS heterozygosity. And the inset shows you the hazard ratio, which is about, again, 0.7, about 1/3 decreased risk, as you can see here. And what you notice here is that the start of decreasing risk on the conversion is somewhere between 75 and 80. And this is kind of contradictory to the previous, might seem contradictory to the previous results where I show the effects between 60 to 80. But if you remember, I mentioned this difference between age at onset and age at diagnosis. So we would expect somewhat a 10-year shift in these findings. So this is also consistent with that prior analysis. And if we again look at the subjects who don't carry APOE4, there is essentially no effect of Klotho VS. Just to show you again across the different studies that we included, there is no heterogeneity in the APOE4 positive stratum. And for the APOE4 interaction, also the effect seems to hold up quite well across the different cohorts. And just briefly to mention, I said at the beginning, there's 9 cohorts for the conversion analysis. What we did is we have actually 7 cohorts across NAC. So here we pulled them together into one cohort because we had very small sample sizes in some of the subsets. So this was to improve the analysis, essentially. And so then we wanted to particularly really replicate or try to replicate this amyloid finding in preclinical subjects. So we focused on cognitively normal subjects between the age of 60 to 80 to really try to be as consistent as possible with this prior study. And also because we think it's a very interesting question, right? If we can find something that reduces amyloid burden in pre-symptomatic individuals thinking towards therapy or preventive approaches, it would be really cool to see a replication of that effect. And so what we see here on the left in blue is the subjects that don't have Klotho VS heterozygosity, and in red the subjects that have Klotho VS heterozygosity, and both of them are APOE4 positive, looking at A-beta CSF values. And so we see a significant difference where it is lower, where it is higher in the Klotho VS heterozygotes, and this is essentially for A-beta CSF, that means lower A-beta burden. And if we look at APOE4 negative subjects, we didn't see any significant difference between the two. And then we looked at amyloid PET as another way to try to validate this finding. This is essentially ADNI that we're looking at here. And so noticeably, we also see this significantly decreased effect here on amyloid PETs, suggesting again reduced amyloid burden. And then in comparison, you can look at the APOE4 negative subjects where we didn't see a significant difference between them. And something to point out here is that if we look at the box plots, it really looks like the APOE4 positive Klotho VS heterozygous positive subjects, they start to look more and more like subjects that don't carry APOE4. And this is similar to what was reported by the prior 2019 paper. And so despite the fact that this was really cool to find this replication, this ends actually the main study that we recently published, we still wanted to shore this finding up more, because even considering all of them together, this was about 700 subjects, 600 subjects. So we set up a multi-site collaborative effort trying to get access to as many as possible pre-symptomatic individuals with amyloid PET scans. So we managed to get together over 3,500 subjects across these 5 cohorts indicated here, where all subjects had an MMSE score above 24, and where we have like nice coverage on different phenotype information on the subjects. And so essentially what I can tell you is that the effect replicates also in this much larger sample. So we can see here the meta-analysis including all 5 cohorts, APOE4 positive, APOE4 negative, and then the meta-analysis excluding ADNI. Despite the fact that here we're looking at amyloid positivity, we have already used ADNI in the prior study. So to really check if it's a completely independent replication effort here, we also ran this extra analysis. And so essentially you see here the odds ratios, and they're very similar to what we saw for the case control and conversion analysis about reducing Alzheimer's disease risk by about 1/3. And you see in both meta-analysis, the effect is significant. And also in this case, we did something extra. We did power analysis to see if in this analysis we were at power to observe this. And yes, essentially we're above 80% power in both meta-analysis. Despite that I'm not showing it, what we saw though in the individual cohorts, if there are less than like 500 subjects in there, we have a quite low power. So in that sense, it was not particularly fully reliable yet what we showed before, but here we have really like shored it up completely. The only thing that we did notice is that when we look at the formal interaction between APOE4 and Klotho, we see the similar effect as we observed before, an interaction of about 30% reduction, but we're not reaching the significance level yet here. But the power for those analyses was also lower. So what we're reasoning or thinking is that if in the future we can add even more cohorts or subjects to this analysis, we're probably going to also see a significant interaction effect here. And so then to conclude on what we actually found, essentially our findings are quite strongly supporting the idea that Klotho VS heterozygosity is reducing AD risk and amyloid pathology in APOE4 carriers. And so here in this plot, which was actually made by Dina Dubal in the cover letter for our article, you can see essentially the plot that they made to recreate our conversion findings, showing the normal increased conversion risk for APOE4 carriers to non-APOE4 carriers, but illustrating that if those subjects then have Klotho VS heterozygosity, the risk of conversion is really substantially going down, and they start to look more like subjects that don't carry APOE4. And these are some of the potential pathways that may be contributing to this. So we have the amyloid pathology work that we're showing, and prior work has been showing also effects on synaptic activity and functional connectivity, that there is essentially increased brain resilience that might also be contributing to this. And so what we also further want to say from this is that for sure with all this work now, it's more and more becoming the case that exploring KL-related pathways for novel AD drug targets or biomarkers can be very interesting. And towards the future, it will be potentially useful to use a combination of Klotho VS and APOE genotypes to refine prediction models to help clinical trial enrichment. Essentially, you want to be recruiting for subjects that don't have Klotho VS heterozygosity and APOE4 because they are more likely to develop Alzheimer's disease and amyloid pathology. And so moving forward towards the future, what we actually are thinking about or looking into, what will be interesting is trying to further understand or validate this effect of Klotho on cognitive and brain resilience, depending on APOE4 status. So in the first place, people have been showing that Klotho affects cognitive and brain resilience, But we do think that it could be good that there will be more studies, because there's also been some heterogeneity. Not all studies are confirming that it's actually having this protective effect. But perhaps by now further investigating how APOE4 plays into that, we'll get actually a better picture. And particularly in that regard, there are some interesting questions asking whether Klotho viscosity affects cognitive decline in pre-symptomatic individuals, and if it differs depending on their A-beta status, and how that associates with markers of brain resilience. And so another very interesting question is looking at how Klotho levels are associated with AD risk. So before I've shown you these different effects, but there is actually a large heterogeneity in these different studies in what they're looking at. And so there's currently no study that asks, if you are Klotho heterozygotes, do you have increased Klotho levels, and do you have, because of that or correlated with that, decreased risk for Alzheimer's disease? So more large-scale integrative studies could really help pinpoint this question and really help answer this question. And then other work that could be interesting to pursue is understanding how Klotho and APOE4 are actually interacting with one another. So there is some interesting literature suggesting that, showing that Klotho actually interacts with the APP processing machinery, where ADAM10 and BASE are actually cleaving Klotho. And then there might be something interesting about longevity-related pathways, as APOE4 is actually known to reduce longevity, while on the other hand, Klotho is known to increase longevity, so there might be some interaction there. And then there's also the potential for the role of inflammation and microglia, because there's prior work showing that Klotho regulates the brain immune interface, and that decreasing levels of Klotho across aging in the choroid plexus actually lead to a more pro-inflammatory state in the brain, which could also then further help seed amyloid pathology. So with that, I would like to acknowledge the people of the lab and all the collaborators. So on the left is Michael Grecius, my mentor and PI, who has really been like a great mentor in teaching me about Alzheimer's disease and making me move forward as a postdoc and making all this work possible. And then Valerio Napoleoni, the lab instructor who recently got a professor position in Italy. He really taught me so much about genetics and really made it possible for me to do all the work I've been doing here. Also, we have access to over 20,000 subjects from more than 20 cohorts, but it's essentially Valerio who, over 4 years, got most of those data together so that we could use them in the lab. Then there's the other members of the lab. And then also within Stanford, we collaborated a lot with Samra Han, who was really helping us a lot with the statistics and very thorough review of the results that we're putting out there. And then other people recently contributed to the replication effort at Stanford, and other collaborators contributing to this replication effort. And so, yeah, with this I'll conclude. If you would like more information, this is the link to our lab. This is my email address. If you have any particular question, feel free to reach out to me. And then I put these two papers here. This is the main paper that I just showed you about the association of Klotho VS with AD risk. And then we have also prior review on the role of APOE in Alzheimer's disease, and what's known since the last 25 years, essentially, on that. And so with that, I can conclude my talk.
Thank you very much, Michael. This was really excellent and very stimulating. So the talk is now up for questions. And while people may think about a question, let me start. So you showed that the protective effect of Klotho is restricted to the APOE4 carriers. And you partially addressed my question, but do you think that this APOE specificity is due to the fact that APOE4 carriers just have a higher likelihood to be a beta positive, and Klotho acts majorly through reducing a beta, or do you think there's another mechanistic interaction between Klotho and APOE4?
Yeah, so for sure, this would be the most prominent way of reasoning that we have about this. Indeed, like APOE4 simply seeds more strongly amyloid pathology, and so that's why we would really expect more an effect of Klotho if it's directly having an effect on amyloid pathology, and less so in APOE4 carriers. But on the other hand, in APOE4 non-carriers, on the other hand, though, in APOE4 non-carriers, then we would still expect some form of effect, right? Because amyloid pathology is still part of the disease. So it's a bit more complex than that, probably. It could be that there are age-specific effects that are also playing a role here. APOE4 is also not only shown to affect amyloid pathology, it might also be affecting cognition in different ways. So in that regard, if there is increased brain resilience via different mechanisms, may also still be that if APOE4 is affecting that there is a more pronounced effect of Klotho VS.
Thank you. So please come forward.
Yes, thank you for the presentation. I have a question. Can you hear me?
Yes.
Yes. So the role of the heterozygosity and homozygosity, can you explain a little bit more? Because I didn't understand quite well what was the difference between the effect. You mentioned the effect, the protective effect was in the heterozygous configuration, whereas in the homozygous was not. And can you explain, or do you have any hypothesis why it's not?
So yeah, although there is limited literature on that, some prior work of Jennifer Yokoyama showed that the Klotho VS homozygotes actually have lower levels of Klotho, even lower than heterozygotes and even lower than the wild types. So what has been shown also previously is that in this case, the heterozygosity effect may be occurring because really depending on having 1, 2, or 0 copies of the allele, 0, 1, or 2 copies of the allele, you may have different effects on secretion. And so when you have 2 copies, your secretion can be again differentially affected compared to when you have 1 copy. So in that way, it can functionally happen that you only have the advantage if you are heterozygote, and you can have even a greater disadvantage when you are homozygote. And so this was shown on Klotho levels, and also that those homozygotes showed reduced brain resilience in the form of intrinsic connectivity. This is also a work from Jennifer Yokoyama.
Thank you.
We have time for a few more questions. So Michael, do you think that the protective effect of Klotho is specific to Alzheimer's disease, or do you think it has a more general effect?
Yes, it's a good question. So some prior work from Dina Dubal or other people have suggested it can also have an effect on Parkinson's disease, for instance. Because it has an effect on cognitive resilience in general, it might just be a widespread effect, excuse me, on dementia. But I don't really particularly know that well in terms of we haven't looked into that, we haven't investigated that, and I think the literature is also not really clear on this matter. So for now, I would be to say that it's pointing toward Alzheimer's disease, and future work will have to look at how it's affecting other dementia disorders.
Mm-hmm. Interesting.
Hi, Michael. Nico here from Michael's lab in Munich. Very nice talk, very impressive data also that you gathered there. I'm also interested in the mechanisms of Klotho. So if it mostly tackles amyloidosis or APOE4 related amyloidosis, are you planning to look also in a more amyloidosis disease model like autosomal dominant Alzheimer's disease? Do you believe that there would be an effect? Because the effect of APOE4 in the B.R.A.I.N cohort, for instance, is pretty small.
But that's actually an interesting question. I have honestly not thought about that yet, but this could be an interesting way of looking at that. But you mentioned that in the B.R.A.I.N cohort, the APOE4 effect is small?
As far as I know, but Michael, correct me if I'm wrong, but as far as I know, the APOE4 effect is not, of course, as big as in the sporadic cohorts, because it's just overrun by the APP and presenilin mutations, I would say. But still, would be interesting to see whether Klotho has an effect here.
Yeah, actually, that's for sure true. And you could also, to some extent, potentially use that to disambiguate if it's still APOE4 specific or not, right? I think in that sense. We could potentially also look at that in some of our data that we have, in the sense that we restricted all our analysis to 60 plus, but we also have subjects that developed Alzheimer's disease before 60. right, is what is covered across neck and so forth, for instance. So we could try to also look in there and see if there is a protective effect of Klotho on those subjects, actually.
And have you looked, for instance, in ADNI at any downstream parameters like FTG PET or even tau PET?
We haven't really explored that yet. We're starting to look into those things, actually.
Okay, interesting. Thank you very much.
Yeah, thank you for the question.
Okay, if there are no more questions, then thank you very much, and thanks to all of you for attending today. Next week's talk will be by Jakob Stern on imaging cognitive reserve, and I would be very happy to welcome you there. Thank you very much. Bye-bye.
Thank you.
Bye.
Bye-bye.
Thank you. Thank you for attending all.